All right, this is the last lecture in our central dogma sequence, DNA translation. The key concept of this lecture will be that translation converts an mRNA message into a polypeptide or protein. A polypeptide is an interchangeable key term with the term protein. This is because amino acids, which come together to form a protein, use peptide bonds to unite them together, right? So they create a polypeptide molecule. So this is synonymous with the word protein. All right, so you might be asking yourself, how does RNA become a protein, right? So there's an actual translation technique that scientists have derived over many, many years, and that is that amino acids are coded by mRNA-based sequences. Translation is going to be converting mRNA messages into polypeptides, which was what we already said. So a codon is a sequence of 3 nucleotides that codes for 1 amino acid. Visually, what it looks like is you have a segment of mRNA, and you are going to divide in groups of 3 each nucleotide. So if we look at the first 3 nucleotides, A, U, G, that is a codon. That is going to code for 1 amino acid. The next 3 nucleotides, C, U, U, are going to code, they're another codon, and they're going to code for a different or equal amino acid. To figure out which amino acid these codons will code for, we need to use the codon chart. Now, this looks daunting, but we are going to go over and break it down, so it's not that hard. So step number 1 down here. If we were, for example, if we had the codon C, A, and U, first we would look for C in the left column. We find it where the red number 1 is. Next, we are going to come over here to where the A is and place our mouse here because it matches up there. And finally, we will look for U. So U is up here, and that leads us to CAU, coding for histidine, abbreviated as HIS. So you might notice a couple of things from looking at this codon chart. There are different combinations of codons, but several codons code for the same amino acid. So the amino acids, let's say isoleucine down here, you can have 3 different codons coding for that same amino acid. You could have AUU, AUC, or AUA coding for isoleucine. Much like that one, there are 3 possible codons that code for a stop. So stop is right here. The 3 codons that code for stop in a gene are UAA, UAG, and UGA. And even though there are 3 codons that code for a stop amino acid, there's only one codon that codes for a start amino acid. Right, and that's methionine, AUG. All right. So now that you see how translation is going to work, you can see how a mutation might throw this off. If the reading frame is every 3 nucleotides create a codon, then if you were to substitute a nucleotide in, you would mess up the entire reading frame. The same thing would happen if you deleted a nucleotide, or if you inserted, if a foreign nucleotide segment were inserted into the mRNA or the DNA code. Right? This is why mutations are so dangerous. However, because several combinations of amino acids code for the same amino acid, this is a bit of an evolutionary mechanism that limits the amount of errors due to reading frame mistakes. So, kind of interesting how that happens. All right, this is another part of the translation complex. Here we have a tRNA. A tRNA molecule is going to have different parts, so it's important to understand what the different parts of the tRNA molecule do. All right, so the anticodon, not to be mistaken with the codon, is at the bottom. It's made up of 3 complementary nucleotides to the mRNA codons. The blue L-shaped body is the tRNA body. And at the very top is the actual amino acid. This circle is going to be detached and left at the top of the rRNA. And this is going to add on to the growing polypeptide molecule during translation. Here is another part of the translation complex. This is a ribosome. A ribosome is made up of 2 subunits. You have the large subunit, which has 3 binding sites, and the small unit, which binds itself to mRNA. Do you see the slip between the large and the small subunit? This is the part in which the mRNA is going to slide through in order for translation to work. So we're going to put it all together now. What is this image telling us? Okay, so on the left of this image, we see a big purple blob. That is the nucleus. It has little pores that you can see, they're blue and black. That is where the mRNA is going to escape from after transcription. That mRNA is going to travel through the cytoplasm until it reaches the ribosome. The ribosomes, as you can see here, are going to slide along as soon as they identify the mRNA. So the ribosome is that green portion of the complex. You can see the large subunit and the small subunit sliding along the red mRNA. You can also faintly see the 3 parts, the 3 binding spots. And those binding spots are going to be for the floating tRNAs. Now, the tRNAs, each tRNA has its own set of anticodons. So in order for the correct tRNA to align in the correct binding site, this mRNA message needs to be read by the ribosome. All right, you can also see at the top of the tRNA that is already at the binding site that you have the growing polypeptide molecule. You see there that the amino acid is clearly labeled and it is joined to another amino acid by a peptide bond. Together, it is a growing polypeptide. If we break this up even further, we have a section of the mRNA, the ribosome attached to the mRNA, and the 3 binding spots in which tRNAs are going to come and go depending on the codons that are being exposed by the mRNA. Those codons are going to be complemented by the anticodons on one end of the tRNA molecules. On the other side, there will be amino acids that detach from their tRNA and attach to the next amino acid, forming a chain of amino acids. Finally, the now empty tRNA molecule is going to exit the ribosome. A complementary tRNA molecule will bind to the next exposed codon, and so on and so forth until we reach a stop codon. Once that stop codon is reached, the ribosome is going to be released and disassembled. That is why the green globular molecule has now detached from its other subunit. The tRNA is also floating freely, and the polypeptide has detached from the tRNA. You can also see that the stop codon is clearly labeled right there, UAG. This is the reason why the translation complex has now detached. And this is the overall product of the central dogma, a polypeptide or protein.